invitation, and it's my pleasure to be here. Today, I'm going to tell you about NAD metabolism and what we are going to do about it for the treatment of age-related diseases like neurodegeneration diseases. You probably have seen this graph before, so we are glad to see that our lifespans increased dramatically, almost doubled since 1900. But as we also see the bad thing happen. As we age, diseases like heart disease, cancer, and other problems become prevalent, and we face these new challenges. In this graph, it also highlights the significant increase of Alzheimer's disease, the most common neurodegenerative conditions, especially after the age of 50s. So there are several hundreds of neurodegenerative disorders, and these diseases are different. They attack distinct region of the brain, as shown in these colorful dots. And also they have different pathophysiology, but they all result from the damage to the nerve cells and the nerve system connections. So these diseases are devastating and affecting more and more people, but the effective therapeutics are lacking, unfortunately, and the progress in developing the drug target specific disease, this progress has been dreadfully slow. So we have this idea that if we target the common problems, we may make the process much faster. So what's the common problem? So here is one. So as we age, our NAD levels decline. This has been associated with various age-related diseases and aging, and also manifest in different tissues. So these diseases are in wide, broad spectrum disease. Also include neurodegenerative disease. On the other hand, if we boost NAD levels, it has a rescuing effect in reducing the hallmarks of brain aging as shown here. I won't go into the details here because previous speaker talked about a lot about these 10 hallmarks of aging, and it also exists in the brain aging. So what's NAD? NAD is actually an ancient molecule. Over 120 years ago, English biochemist Arthur Harden discovered it as a cofactor in fermentation, and it caused Alzheimer's. Later, Sweden biochemist Hans von Euler-Chelpin studied its chemical nature and action. In his Nobel lecture, he emphasized its significance and saying it is one of the most widespread and biologically important activator. Now we know that NAD doing much more than helping fermentation. First, it's essential critical redox carrier in metabolism. So it's a cofactor for hundreds of metabolic enzymes by converting this NAD plus to NADH. And it's also referred to as energy metabolite because it plays a critical role in energy metabolism from glycolysis to TCO cycle and the oxidative phosphorylation. So now we know that the second function of NAD is it's a critical signaling molecule in many signaling transduction pathway. And this function mainly through four major NAD consuming enzymes like sirtuins, PARPs, and CD38 and SAM1. So it regulates various cellular processes like metabolic homeostasis, stress response, aging processes, and also the DNA damage and programmed cell death, calcium signaling, inflammation, and exon degeneration, etc. So the first link between NAD and the neurodegeneration came from the genetic study of injury-induced neurodegeneration called Wallerian degeneration. The German physiologist Waller observed myelin sheaths disrupt, fragmented in axon after distal to the injury site after nerve crash. And then later, a mutant called Wallerian degeneration slow was found. And this mutation involved overexpression of NAD as a synthetic enzyme called NAMPT in cytosol. Just like the name indicated, the Wallerian slow mice survived showed prolonged axon survival after nerve injury, and the mouse model showed protection in many animal models of neurodegeneration. The further evidence came from forward genetics in fly. So there's a genetic screening for the mutant resembling the Wallerian slow phenotype, and 3 mutations were found. Amazingly, all 3 genes lies in one single gene called SAM1. And this SAM1 turned out to be the NAD hydrolase, and the activation of SAM1 triggered the exon degeneration program. So based on this knowledge, SAM1 has been a promising drug target, and several SAM1 inhibitors already developed. So I just told you that NAD metabolism is a promising drug target. So in our body, how cells make and use NAD. So there are 3 pathways to synthesize NAD. The salvage pathway, press handler, and the de novo production. And in our body, this salvage pathway is the main source of the NAD. So in this pathway, the nicotinamide and the PRPP is combined by this rate-limiting enzyme called NAMPT, and followed by the formation of NAD catalyzed by another enzyme called NAMPT. So the rate limiting, what does that mean? That means that the speed of the enzyme determines the NAD synthesis speed. And you can also see that all 3 pathways don't need the NAD enzyme. So I just mentioned the NAD consuming enzyme. So these 4 class of NAD consuming enzyme break down the NAD, generate nicotinamide, and nicotinamide return to the salvage pathway. So based on this picture, so I think the audience already figured out then how we boost the NAD. So there are 3 strategies. The first one, you can take supplemental NAD precursors like NMN and NR. So many people have already tried that. And the second one, inhibiting NAD consuming enzyme like SAM1, CD38. The third one is activating key NAD biosynthetic enzyme like NAMPT and NAD. And our lab focus on the third strategy. So how we dive into this NAD field? So we actually go in this by a very serendipity discovery in Steve McKnight's lab at UT Southwestern. So they did a very brave phenotypic drug screen in mice, looking for the chemicals which can promote newborn neuron formation in adult brain. So sure enough, they find one and named the P7C3. So the P7C3 analogs, active variants, showed remarkable activity in many neurodegeneration model systems like LS, Parkinson's, and traumatic brain injury, Alzheimer's, and chemotherapy-induced peripheral neuropathy, CIPN. So the big question is how this small molecule can do so many things. Later, we found that the P7C3 targets NAMPT, the enzyme we just told you, the rate-limiting enzyme for the NAD salvage. And in the situation of cellular NAD depletion, the P7C3 activate NAMPT and enhance the NAD level, cause the NAD rebound. So going from the phenotypic drug screen, we then carried out a target-based drug screen, looking for the chemical activator of NAMPT. So generally, it's very easy to get an enzyme inhibitor, but very difficult to get enzyme activator. This is the same thing. It's true in our case. So from 50,000 chemical library, we only found several chemicals can activate NAMPT. And we pursued the most robust one we call the NAD. And from here, we can see that NAD can robustly activate the NAMPT and make the enzyme produce NAD faster. Consistently, in the enzyme kinetic assay, we see that the NAD can increase the maximal velocity of the NAMPT enzyme. So then, how does NAD binds and activate NAMPT? So we solve the crystal structure of NAMPT in complex with NAD. So the native NAMPT is in a dimer formation, so as shown here, the yellow and the pink. The NAD chemical actually sit at the tunnel, which is the active site reside. So NAD binds in a big binding pocket here, filled with water, and some residues also can independently bind to NAD. So we mutate these important residues, mediated the binding, and choose the mutations which doesn't affect enzyme activity. And we see this mutation no longer be able to be activated by NAD. So how does NAD activate nicotinamide, activate the NAMPT enzyme? We did a structure alignment. So the NAD actually locate a little way, like slight away from the substrate binding site. Doesn't overlap, and the NAD doesn't cause the change of the overall structure of NAMPT. But at the binding site, there are conformational change, especially this phenylalanine 193. The sway of the phenyl ring push the nicotinamide closer to PRPP, make it the condensation reaction easier. So we think, we propose that NAD is an allosteric activator of NAMPT. To improve the NAD chemicals, we collaborate with Professor Tang Feng to optimize NAD. After many rounds of structure optimization, and we got a more active one in both binding activity and the enzyme activation activity. So we call this variant NAD5R. Based on the docking model of the NAD5R with NAMPT, we see that this siren group directly bound to the lysine 189 residue, make the binding more tight. So after seeing all this NAD binding to NAD and stimulate the enzyme activity, we ask if NAD can enhance the salvage flux in cells. So we did this tracing experiment with the C14 labeled nicotinamide in HepG2 cells. From this C-layer chromatography, we can see that both NMN and NAD was increased. So how about the energy metabolism? We did the metabolic analysis and seeing that the nicotinamide was enhanced, and so does the T cell cycle and TCA cycle and glycolysis fatty acid oxidation. How about the oxidative phosphorylation? Using the seahorse to measure the oxygen consumption rate, we can see at 1 hour treatment, NAD caused the dose-dependent manner of increase of basal and maximal oxygen consumption. But at 3 hours treatment, this trend disappeared. This means that NAD can cause dynamic remodeling of energy metabolism. How about the original purpose of neuroprotection of NAD? So we use neurostem cells models, and when we restore the growth factors, FGF, EGF, so the neurostem cells quickly differentiate and die. But for the NAD and the P763 chemicals, they not only increase the stem cell proliferation, but also inhibit the differentiation of the stem cell, like shown here, like the neuron marker QJ1 and the astrocyte marker GFAP. So as a proof of concept, we test the efficacy of NAD in a mouse model of chemotherapy-induced peripheral neuropathy. So as the CIPN actually is very debilitating disease for the treatment accompanied with the cancer treatment, and the cancer drugs like paclitaxel and Christine cause this severe side effect. Using this model, we can see compared to the paclitaxel treatment, the NAD treatment can improve the pain sensation, maintain the mining integrity, and keep the nerve conduction speed. So as I mentioned that two enzymes are important for NAD biosynthesis. This is including NMNAD. And the NMNAD has 3 isoforms in different cellular compartments. NMNAD2 is in the cytosol and and mainly expressed in brain. So we did a similar strategy to our second target, NAMPT. Surely enough, we did a similar screening and found the robust activator of NAMPT we called NAMPT. And this NAMPT also showed a neuroprotective effect in the CIPN models. So in summary, NAD plays a very important role in promoting neuronal health, and based on this knowledge, we have discovered 2 types of small molecule targeting NAD biosynthesis to boost NAD. So both all these compounds show the neuroprotective activity, demonstrating the potential of activating NAD biosynthetic enzyme as an approach to treat neurodegeneration. So currently, we are extended to evaluate the longevity impact of these activators in mouse model. So I would like to thank the forum because it provides valuable insights for the biomarkers and the aging clocks for us for our assessment. So finally, lastly but not least, I want to thank the amazing students and postdocs in my lab. They have worked very hard for the last 5 years. And thank for our collaborators and funding agencies. Thank you all for listening to my talk.
感谢王科林老师的分享，下面是提问时间。
Excellent talk. Can I ask you if you boost NAD or whatever related to NAD plus stuff, which organ do you think will benefit the most? And which organ do you think will benefit the less? Based on your theory.
If I give the guess, I think probably the neuronal tissue probably benefit the most. I'm not sure. I never did a systematic study on it.
Hello, Professor. Thanks for your speech. And I focused on you just mentioned NAMPT and PT. And so I also read some papers about the NAMPT. And they think that NAMPT is a kind of risk about cancer. So what do you think about this thing?
So this is a good question. So actually NAMPT is served as a drug target for a long time. At the beginning, it was a drug target for cancer treatment because some cancer cells has more active NAMPT. That's the rationale. But all these trials failed because of the side effect of NAD depletion. So the scenarios the important question is when we need to boost NAD and how much we boost NAD. So when we age, when we have the degeneration, when the NAD depleted, we do that. I think that's the way. And also, many people already taken the NMN or NR. I didn't hear any cancer side effect from that.
But one more question, because I have been eating NMN for a long time, but...
You have?
Yeah.
You don't need it.
That's kind of my habit. But I found it and noticed they have a lot of different kind of NAD supplements such as NMN, NR, and also NADH and NMNH. So how do you think about the difference between all the supplements?
I think for this supplement, I think the drug metabolism, the PD profiling should be very critical. So I can't give you the answer. We never look at that. And that's the thing. I think it's very critical to figure out by scientific method. I think Joe will tell us that's very important.